104, we will examine organisms within the domain Eukarya. We are now going to examine another supergroup of eukaryotes called the Archaeplastida. These include many photosynthetic organisms that are important in forming the base of many terrestrial as well as aquatic food webs. These consist of red and green algae, both unicellular and multicellular forms, as well as plants that it is believed originally descended from green algae. These organisms all contain plastids and are believed to have descended from an ancestral heterotrophic protist that originally engulfed a cyanobacterium. Take a look at the phylogenetic tree below to understand the lineages in the Archaeplastida group. We have red algae, green algae, which consists of both chlorophytes and charophytes, as well as plants. We are now going to take a look at red algae, which is also known as rhodophytes. Red algae gets its red color due to the presence of the photosynthetic pigment phycoerythrin. This pigment can mask chlorophyll, giving it its red color. However, varying pigment levels between phycoerythrin and chlorophyll can cause some to look greenish in shallow water or black in deep water. Because some can absorb blue-green light, they can be found at very deep depths. Red algae is very abundant in coastal areas in tropical oceans. However, it can also be found in some freshwater and terrestrial environments. In addition, some species can be heterotrophic and parasitic on other red algal species. Red algae typically reproduces sexually. However, due to its sessile nature, it uses the water currents in order to bring the gametes together. Red algae can take on many multicellular forms, and it can be filamentous as well as leafy. A common example of red algae is nori, which is the common seaweed found in sushi. Green algae gains its green color through the presence of green chloroplasts. Green algae is considered to be paraphyletic and contains two main groups, the charophytes and chlorophytes. Charophytes are the algal species that are most closely related to plants, while chlorophytes are considered to be more distantly related. Chlorophytes consist of both unicellular and multicellular life forms. Some can be solitary or symbiotic with other eukaryotic organisms. They can be present in freshwater, marine, and terrestrial environments, and in some extreme environments as well. They reproduce using sexual and asexual reproduction, and some species may also have the alternation of generation between the haploid and diploid forms. This diagram depicts the typical life cycle of Clematomonas, a unicellular chlorophyte. Note that the haploid part of the life cycle is depicted in blue, while the diploid part of the life cycle is depicted in pink. Also note that both asexual and sexual reproduction can occur. If we begin with a mature cell, the mature cell exists in a haploid state. If harsh conditions arise, the mature cell can produce gametes, both positive and negative. These gametes represent different mating types that can eventually fuse and undergo fertilization to form a zygote. The zygote can secrete a hardened shell which will help it withstand harsh conditions. When more favorable conditions arise, the zygote can undergo meiosis to form 4 haploid individuals. These will eventually emerge and mature into a mature cell, once again in the haploid state. Under more favorable conditions, the mature cell will typically undergo asexual reproduction pathways. This means that the mature cell will undergo mitosis to produce different mating types. It will also typically resorb its flagellum in the process. Once those individuals are released, they are called zoospores. The zoospores will have individual flagellum that will help them move within their environment. These zoospores will eventually mature into a mature cell. Green algae offers a unique perspective to understand the development of multicellularity. Many green algal species have an increased complexity due to the formation of colonies, the formation of truly differentiated multicellular bodies, or they may have the repeated division of nuclei, leading to the formation of multinucleate supercells. We're now going to take a look at the last supergroup of eukaryotes, the Unikonta. These are also called amorphia. They're typically represented by two main clades, the amoebozoans and the opisthocons. The amoebozoans include amoebas with tube as well as lobe-shaped pseudopodia, while the opisthocons include present-day animals, fungi, and other non-amoeboid protists. This classification system is primarily based on molecular similarities, such as examining myosin proteins, genes, as well as whole genomes. The root of this particular tree is currently under debate and not certain. However, recent theories suggest that unikonts actually arose closer to the common eukaryotic ancestor. This indicates that the root of the tree exists somewhere between unikonts and other eukaryotes, leaving Excavata, Sar, and Archaeplastida forming a clade. The first group we will examine of Unikonta is the amoebozoans. These include three main groups: the tubulinids, slime molds, and entamoebas. The tubulinids are identified due to their tube or lobe-shaped pseudopodia. Many of these are present in marine, freshwater, as well as terrestrial environments. Most of these organisms are heterotrophic and prey on bacteria and other protists in their environment. Slime molds typically produce fruiting bodies to disperse their spores. Though this is similar to fungi, DNA analysis suggests that this arose and is an example of convergent evolution. Slime molds are typically represented with two lineages: plasmodial slime molds that result from large multinucleate unicellular masses called plasmodium, and these extend to pseudopodia to engulf food, or a cellular slime mold. These exist as solitary cells that can form. Slime molds typically produce fruiting bodies to disperse their spores. Though this is similar to fungi, DNA analysis suggests that this is not the same, and that these abilities arose through convergent evolution. Slime molds are represented by two lineages. Plasmodial slime molds typically form large multinucleate masses, called a plasmodium. This mass can extend out as pseudopodia to engulf food. Cellular slime molds, on the other hand, are solitary cells that can form a multicellular slug-like aggregate during adverse conditions. This slide here depicts an example between a plasmodium slime mold and a cellular slime mold in its aggregate form. This diagram depicts a typical life cycle of a plasmodial slime mold. Note the difference between the haploid form in blue and the diploid form in pink. A zygote will form a feeding plasmodium that will eventually mature and prepare or produce fruiting bodies. This fruiting body will exist on a stalk and is termed the sporangium. As the sporangium matures, it will produce spores. These spores are considered to be haploid. These spores will then germinate and produce flagellated and amoeboid cells. These cells can undergo fertilization to then form a zygote that will then mature, forming a plasmodium. This slide here depicts the typical life cycle of a cellular slime mold. Note that the haploid state is depicted in blue while the diploid state is depicted in pink. We begin with a solitary amoeba, which is in the active feeding stage. Also note that this amoeba is considered to be haploid. These haploid individuals can mate and undergo fertilization to form a zygote. The zygote is considered to be diploid. This diploid zygote can then undergo meiosis to produce more amoebas. Also note that the solitary amoebas can undergo asexual reproduction, whereby they divide through mitosis. However, when resources become scarce, an interesting behavioral and multicellular formation can occur. Many amoebas can gather together and form an aggregate. This aggregate can then move along and migrate to other locations. However, it only migrates for a short period of time before it stops and produces fruiting bodies. These fruiting bodies are produced by the differentiation of different amoebas. Some amoebas will then migrate to the tip and eventually develop into spores. These spores will then be released and they will then develop into amoebas themselves. The entamoebas are considered to be symbiotic parasites of vertebrates as well as invertebrates. For instance, in humans, Entamoeba histolytica is considered to be pathogenic and the main cause of amoebic dysentery. This parasite is easily spread through contaminated water and food sources and is known to be the third leading cause of death due to a eukaryotic parasite. The last group of unikonts is the opisthocons. This is considered to be a highly diverse group of eukaryotes that includes protists as well as animals and fungi. One of the reasons why the kingdom Protista no longer exists is our recognition that protists arise many times in evolutionary time. For instance, euclarids are considered to be more closely related to fungi, while the choanoflagellates are considered to be more closely related to animals. Thus, the evolutionary tree has been revised. We know that protists can play key roles in ecosystems. Though most protists are considered to be aquatic, they can also be found in many different habitats. Protists are very common in oceans as well as ponds and lakes, and they can be also found in moist terrestrial habitats. Many protists are considered to be important symbionts and primary producers. Many protists are considered to form symbiotic relationships with different organisms. For example, some dinoflagellates are known to provide nutrients to corals and can play a vital role in coral reef formation as well as diversity, whereas some protists are known to inhabit the guts of termites to aid in the digestion of cellulose in wood. Some protists are also known to be parasitic, such as Plasmodium, which we already examined, that is known to cause malaria, Festria chamoyii, which is a dinoflagellate that is known to cause massive fish kills, and Phytophthora ramorum, which is a stromenopile, which is known to cause sudden oak death. Some protists are known to be photosynthetic and can utilize light energy to convert inorganic carbon or carbon dioxide into complex organic compounds. As a result, these species form the base of most food webs. For instance, in aquatic environments, protists are known to comprise 30% of the food web base, along with prokaryotes and plants. Given their role at the base of the food chain, small changes in producer populations can have large-scale changes in the rest of the surrounding community, both aquatic and terrestrial. In many aquatic environments, producers are limited by nutrients in their environment, such as nitrogen and phosphorus. Nitrogen is commonly considered a runoff from agricultural processes. When nitrogen seeps into the water table and eventually seeps into a body of water, this can cause nitrogen to no longer be a limiting element or nutrient. As a result, this can lead to population explosions or algal blooms. Climate change can also have an impact on primary producers. Though climate change has been known to increase sea surface temperatures, this has been followed by a decline in many of our photosynthetic protists and prokaryote species and organisms. It is believed that this is due to a decrease in the movement of water, leading to less upwelling of cold, nutrient-rich waters from below. As a result, these nutrients are trapped lower in the water column and do not reach the higher areas of the water column where most of these photosynthetic protists and prokaryotes reside. If this trend continues, this could cause major changes in many marine ecosystems as well as fishery systems and can ultimately affect our global carbon cycle. The next two slides give you an overview of the supergroups that we examined today: the excavata, sarclade, Archaeplastida, as well as Unikonta. Please take a look at these slides to get an overview of the major groups we reviewed, as well as the key characteristics and some examples.